everyone. So today we are looking into art. So basically, it's an art day as usual, the normal trend. And so Brian Pondi here, and then weekly from Agumba Oluoch, who's doing a PhD at University of Bonn. We also show some of his projects using R. And yeah, but for now, I'll just be giving an overview, an introductory session on why R or is R worth it to look into, or maybe it's really underrated. You can never know by some people, especially there's usually this debate of R versus Python and stuff, right? So basically, yeah, an intro. We'll skip that. Like people usually tend to know each other every time and then. Right now, we're going to look into why R, then R or Python. Then for my case, I'll just share the resources, and then after that, Wycliffe will take over. Okay, so the question is why R and GIS or why R and Geospatial, and yeah, the first thing it's usually there's the large community, right? If you use a tool, you definitely usually need proper backing of a community so that when you get stuck, you can easily find, it's easy to find solutions, especially because the large community tend to share a lot, right? The second thing is that R is usually made open source, and it's free. You don't need to pay for licensing to use it. Yeah, and for anybody coming from like a background of Python, also R has got the packages or the libraries, so they're known as CRAN packages, and there are tons of libraries or packages to use, from ones that contain data sets to the ones that have statistics concepts, to ones that you can use to fill gaps in satellite images, among many more stuff. Another thing is that there's definitely contributes to fast productivity and also automation. So, for example, if you're doing if you're using the same methodology in multiple areas, so once you write down the scripts, it's just about the input that will change, which is the data sets, right? And yeah, that's a main great bonus. Yeah, so R was mainly built for statisticians. So if you ever think about statistics, you can never go wrong using R. There's usually this joke about if you're using R, you don't ask if it's possible, you just ask how, how to do something on matter statistics. And also right now there's key drive about reproducible research. It's now in the trend that if you do research, then if someone gets your data sets and your R scripts, are they able to run the scripts and get the same output? So that's what reproducible research is all about. Maybe other people to validate your stuff. Yeah, and so another key one is that this is a transferable skills, right? To data science and analytics. Yes, we focus so much on spatial data, but then data is data. And once you know something like R, because it's mainly R and Python that are usually the most popular languages in analytics and in data science spectrum, right? And so R kind of started picking up in the spatial data space probably from 2008 when this book came out. They applied spatial data analysis with R by Roger S. Bivand, Edza Pebzema, and Virgilio. Yeah, it was quite like when the book came out is when people started absorbing and using it, because these guys were like the main contributors of our spatial packages. And so the question is usually Python or R, or which one is great, right? And there's usually like even a clash of communities where Python guys see themselves better than R guys, and R sellers see themselves as better than Python guys. And the best advice I can give here is usually to know both. But then again, it depends on what you really want to achieve, right? Or in which kind of area you are working with. And so if you look at differences between R and Python, so R was mainly developed to cover statistical modeling. So it was mainly built for statisticians, and hence you can really never go wrong with that when you're doing tons of statistics modeling, right? Python is a multi-purpose language, definitely was built for automation, web application development, and data analysis. And so for me, the fact that I've interacted with both, I've kind of realized at times I usually tend to have way more statistical tools compared to, for example, Python. And maybe this is because most researchers maybe use R. Yeah. And then, yeah, here's where we used by, so it's mostly statistician, analysts, and data scientists use R. In Python, it's developers, data engineers, and you can say data scientists. Also, the data analysts, definitely, yeah, they share, right? A little bit, but you can see R is statistic heavy. Okay, so yeah, what I also realized, if you compare Python and R, who is suitable for, R is usually friendly to someone who doesn't have any programming experience, because it is way simple and easy to pick up. Compared to, for example, if you are doing Python, yes, it's also great for beginners, but it also has got these things to do with the object-oriented programming, solid principles, among others, that aren't really properly there in R. I can say, yes, it's Python is more, if you're from a more software engineering side of things, Python might look interesting and catchy for you to understand compared to R. But if you look at R as a general user, not to the software engineering mindset, you will find that it's easy, kind of. Like, for example, when you try to write the script, we just write and define them to a variable, and to a variable, so it's like a step-by-step process of writing things, or procedural. Okay. And the main package distribution in Python, in R is cran, while in Python is the pip or pip install, if you remember what you usually do. Yeah, then visualization tools, examples in R, ggplot, plotly, gggraph, in Python, matplotlib, bokeh, seaborn, among others. These are just an example. The more you tend to use these languages, the more you tend to discover more tools and resources and what to use depending on your problem. Okay, so yeah, I'll just mainly give like 3 resources. It was kind of a workshop, but it's well done and posted online for beginners. Modern geospatial analysis with R, and then there's geocomputation with R, and then spatial data science with R. If you kind of utilize these 3, I believe it helps to greatly pick up R and utilize it for spatial. And yeah, you can also specify YouTube, Google, among other resources. Actually, R has got way tons of resources than you can ever imagine. So to simply show you, for example, these links, we can start here. So for example, the modern geospatial data analysis with R. If you look at it, it basically covers what a really beginner needs to know: the introduction, reading in spatial data, mapping, coordinate reference systems. These are key. Deeper dive into vectors, deeper dive into rasters, geoprocessing with vectors and rasters. So this is really easy. And if you look, for example, in R, if you're reading vector data, after importing the SF package or installing it, it's just like read, then pointing to shapefile, or read SF, GeoJSON, or read SF, then geo package, and this you're assigning into this variable borers. Yeah, it's something you can easily check about, so we don't really need to go deep into the basics. And yeah, if you come to also raster, this raster package. There's also another one called stars, but yeah, it's library you raster, and you approach the canopy, you can see the raster. And there's also a break part of raster package for multi band reading datasets for more than one band, and you can plot, and that's kind of pretty straightforward and easy to pick up. That's why it's really friendly to newbies. The second stuff that I recommended is this spatial data science with R. It's quite interesting, also written by the initial authors of the analysis of spatial temporal data with R. And this is Edzap Ebzema and Roger Bivand. And if you look, it also has got getting started, reading a fast map, and visualizing, right? There's about coordinate system, geometries, manipulating data, and so forth, going up to, I think, geostatistics, there's interpolation and geostatistics. So it's quite interesting stuff, right? And then the final one here is now the geocomputation with R. So this is also freely available online, and yeah, these are actually great resources for one to get your feet dirty, because they've got sample data sets you can play around and just, yeah, the master R. And I think that's it for this. And so for now, I'll just pass over for Wycliffe to do the hands-on, to show some stuff, interesting stuff. Wycliffe.
Thanks, Brian.
Yeah, okay.
You allow me to share my screen.
Yeah, stop sharing. There, you are allowed.
I hope that is visible across the globe.
Yes, it is.
Thank you so much. I'm Wycliffe, as Brian has introduced. I enjoy species distribution modeling in R. The work I'm going to share here is by myself and coding in Geo members. So you can see the authors up there, Wycliffe, and coding in Geo members. So it's for all of us. Today, the 21st of February, 2021. Well, species distribution modeling is all about questioning where could a species occur over space, given occurrence records that we already have, as well as environmental variables that are responsible for the observed occurrences distribution. So here is a view of what is happening. Like you can see on my left-hand side, we have several environmental layers down here. This could be rainfall data. This could be soil pH data. The upper one could be temperature data. And then somebody also walked into the field, and in the same field, in the same area, and observed a species X somewhere, species X somewhere, and then no species X somewhere, and no species X somewhere. So what this person is doing now is to say, where I observed this species, the temperature was this, the soil pH was this, and moisture or whatever other variable was this. So these three variables occurred or were observed where the species was recorded. So it means if we do that across space, if we do it for several locations, then we can have environmental variables with some confidence that these are the environmental variables which are determining the distribution of species X over space. So what we do with the occurrence records and the environmental variables is to throw them into a modeling framework where we have a range of environmental variables and how a specific species is responding to their changes over space. Like where the temperatures are low, where the temperatures are high, how does that species behave? So from that, we can generate layers of, we can generate output or model outputs that depict the distribution of the species. But sometimes the models can be so many. You can have RF, random forest, you can have support vector, you can have general linear model, you can have so many models predicting the potential distribution of the species. And then if you take all of them to the policymakers who want to, who are concerned with the species, they will ask you, so which one do we use? Yeah, so there are some model evaluation steps that you go through before you pick a few or selected few of the models to generate an ensemble model. So this is probably the last output which you show to the policymakers or those who are concerned with the species. So the whole process starts with overview, which is about conceptualization. You think about the species of concern, if it is Panthera leo, Panthera pardus, or whichever species. You think about it in and out. You read a lot about it. You get to know the species fully before you think of getting biodiversity data, partitioning your data into training and testing, as well as the environmental data, and then even transferring of the data of the model to other scenarios or other geographic regions. Then the third step is model fitting, where you select your variables. You can have so many environmental variables. The layers of environmental variables are kind of infinite, you may say so. But then those that are responsible for your species of concern distribution might just be a handful. So those are the ones you use in fitting your model, and even up to threshold selection, where you say, if the model output probability is up to this level, then I say there is a presence there. But if the model output probability is below that, then we say the species is not there. Then assessment, you can perform some statistics assessment of the performance of your model, as well as using expert judgment. You might model the distribution of a lion using your environmental as well as occurrence records. And then you find that there are a lot of lions on Lake Victoria. So it needs expert judgment to tell you that, hey, this species cannot occur on top of Lake Victoria. So you go back to your model and say, okay, so is there an island in Lake Victoria where there are plenty of lions? So those needs expert judgment. And then predictions, you can make predictions once the expert judgment and response shapes are fine. You can make predictions using your model. But if that fails, then you go back to the cycle of conceptualization. Think again about the species, think of the concerned environmental variables, data sources, and all those, and you do the fitting again. A number of researchers have published books, and even papers are plenty about species distribution modeling. Others call it ecological niche modeling, some habitat suitability modeling, some climate envelope models. So there are so many names which are attached to almost the same thing, but if you dig deeper, you find that species distribution modeling is not ecological niche modeling. Well, we have this first book, Mapping of Species Distribution by Janet Franklin, a very good book giving a great background on the topic. Ecological Niches by Townsend and his colleagues is also great in theoretical background. How do we think about environmental variables and occurrence of species, and then use that to predict or project the distribution of a geographic space? They call it Hutchinsonian duality. Then we have Habitat Suitability Distribution by Antoine Guizan. It's another great book. This is specific to R, same as Joint Species Distribution Modeling. So species distribution modeling started by largely looking at individual species. If you take this fox, you look at the fox alone. If you take a specific grass, you look at the grass alone. But then somebody said, okay, if, for example, you want to build an industry somewhere, you are not only concerned with a specific species. You might need to know several species within the same locality, and therefore joint species distribution modeling has arrived. The framework which I showed in earlier slide was published by Zurel and others, around 22 of them, of authors, to try and make the work reproducible, so that if you do your ecological niche modeling of species X, and then you throw the same information to somebody to run the same model, they get similar results. Then SDM package is the package we are going to go through today. It was written by Naimi and Araujo. It's a great platform. What was in the mind of Naimi was reproducibility, and also making the package extensible so that you can alter some specifications within the package to suit your need. So without further ado, we will move to a demo. My art studio is visible.
Not yet, we're still on the slides.
Are we able to see? Okay, I'm still on the slides.
Yeah.
I need to stop. Share that. Okay, share screen so that changing from one to the other is easy.
Yeah,
Not
visible.
yet?
Yeah, visible now.
It's okay now.
Okay. Thank you. Thank you so much. Well, the task ahead of us is species distribution modeling, created on 19th, and I've just updated it today, this morning, by me and you. Then the main package we are using is SDM. Version documentation, should you run the same script and find some problems, then probably it's because you're using a different version of one of these. So I'm using R 4.03. 4.04 was released 1 week ago, about a week ago. RStudio 1.41103, SDM, and the rest are indicated there. So if you have a different version, then you know why it's working or not. Library SDM. So in R, what we do is that we load libraries using library. Some other programs you can use, is it import, something like that in Python? Java, I have no idea. JavaScript, I have no idea.
Yeah, it's import.
Then, thank you. Then, so library SDM, if I run that code, I can run it using this run here. You can click on that run and it loads safely. I'm not going to run this install all. It's only if you are running library SDM for the first time. So maybe the best thing is to comment it out. Then library Dismo. Library SDM is what is having the functions which will help us to build the model. Library Dismo is giving us get data function to access some occurrence records. Then library tidyverse is for wrangling the data. You will see that. Map view, I think it's map view is for displaying the outputs using global tiles. So those are the tiles. Then we have library USDM, which will help us in collinearity tests to remove collinear variables and remain with those that are not collinear. Gravia tenax is the species we are going to model today. It's a plant species, mostly terrestrial. And I like it because I ate it when I was young in primary school. At the same time, we used it to cover books. How? Okay, the fruits are a bit gummy, so we could use it to stick the kind of papers, the newspapers, to cover our exercise books way back. Then, so it's of interest to me. The fruits are sweet. Species occurrence data we will get from GBIF, Global Biodiversity Information Facility. I hope we are familiar with it. If not, we can click on that link and see what it's all about. There are also iNaturalist as a source of occurrence records. These are where people who have gathered data from the field occurrence. I saw a lion here. This is the coordinates. This is all the information related to the occurrence there, and you throw it to the database so that other people can use it to run a range of projects. VertNet is also another one, principally having vertebrates, so including humans, occurrence of humans, but also apes, birds, those are vertebrates. Bien, biological information, biodiversity information and ecological network, something like that. It's also having some information on occurrence records. You can also walk into a museum and see some preserved records, preserved specimen, and you find some location on them and you extract that for your fieldwork. Field excursions, you can also walk into the field, see a species somewhere, pick the record. Climatic or predictor variables we'll get from WorldClim. So WorldClim again has got present, future records of climatic variables, a range of climatic variables across the globe. I think not in Antarctica, but tropical Africa, most parts of the world are covered. There are also other sources of occurrence records, AfriClim, you can see that. If you are specifically modeling within Africa, the AfriClim is better suited than WorldClim. RCMRD, they also have GeoPortal where you can get some environmental variables, especially if you're working in Eastern Africa. NASA, NOAA, drone, if you can fly drone over space, gather environmental variables, you can throw them into your model. NEON is also a source where you can get this data, humanitarian databases. Some of them you might know can help you get environmental variables to throw into your model. You can also work with soil auger into the field. So you see a species and you sample the soil there, take to the lab and do analysis, see the pH values, see the whichever values, metals values, concentrations of whatever you are interested in, and then use that as a layer. Not so much recommended because all other places you didn't access, you have no idea. But if you use the remote sensing or AfriClim or RCMRD or whichever other resources or environmental variables, then you are better placed. Well, so I will call this my species Grevia. Sorry, I'm using Grevia because of the German accent of W. Otherwise, it is Grevia tenax. So for us, for majority of us, I think we'll call it Grevia GBIF. Then GBIF function is from this small package. Then we provide argument genus, feed it Grevia, and then the species tenax. Download true, n tries 10. So genus is the genus of the species, Grevia, and then species is the name, species name, which is tenax. Then download T means we want to download the data. So if we run the code, it will, if we run the code, it will download the data. N tries 10 provides the number of tries which this function will throw into the database. Like query that database 10 times. If you still can't get the data, forget about it. Sometimes these databases are busy. Like I think the other time we were talking about Google Earth Engine, somebody said it's better to work with it while the other side of the globe is sleeping. So again, with GBIF, you are better off give it some number of tries. I think the default is 1. So if you try querying a GBIF and you don't get results, then it means it's many people are querying the same database at the same time. So you can give it 10 tries. So it will try 10 times. You can increase it to 20, 30 until you get the data. But again, if you're trying several times and not getting the data, then probably that species has got no records in that database. Then Kenya extent, what can I show here? For example, if I say download is false and I pick this and run, what you will see is 859. So it tells me that it has queried the database 10 times or maybe once, because if it gets the data once, it doesn't need to go to the second one. So it has queried the database and found 859 records of this species. If I remove the species name or the species argument, then it will query the whole of the genus. So any genus of Grevia will be returned. So you see the number of individual records goes up to 56,000. So there are several species within the Grevia genus, but I'm only interested in Tenax for now. So I downloaded this earlier. I will not download it now because it's in my R environment. So I don't need to download it again. Then Africa extent, Africa EXT. So that is the extent of my study area. I'm interested in modeling the distribution of these species within Africa. So this is the minimum longitude. This is the maximum longitude. Negative 38 is the minimum latitude, and the maximum is positive 36 above Tunis. Then, so this can help you to crop or clip your study, your environmental variables. So because I already saved that, we can check the class. It normally is not found. Wow. So it means I will have to run it again. Fortunately or unfortunately. Again, when downloading this, it runs in chunks of 300, 300. So from 0, you will see 300, then 600, then 859, done. So 300 records downloaded. It's taking more time than I expected, 600 done. So the remaining 259, please. Good. So we can check the class. It's a data frame. I don't know whether we have data frame in other databases, but it has got rows and columns like a spreadsheet. Those who are using other programs, you have a better name for it. We can check the first 6 rows. Other programs, I think it is, if you use head, it will be like the first 4, 5, and then the last 5 separated by some dots. But here it's the first 6 rows. And the first row, which we call observation, is number 1, not number 0. Those who use programs having number 0 as the first observation, no. Then, so you can see we have so many, it has thrown a lot of information on my console. I don't need all this. And if we check dimension to know how many rows and columns it has thrown, then we see 600 rows and 146 columns. Not all information which is in the database is good for use. People throw in trash as well as important data there. So one way to check that is using this function table, and then we table the column basis of record. So this will group the counts of individual observation basis of record. Like human observation in my database are 28. These are the observations of Grevia tenax or Grevia tenax that were done by human beings. Living specimen that were seen, again, are 11. Material sample, these are like you saw Grevia tenax in a lorry passing by or something like that. So it's not a living specimen you found somewhere growing. Preserved specimen, these are from museum and such like institutions. And then unknown, there is another one called unknown. So you will not be interested in using unknown record to run your model. nobody knows how it was recorded. So to clean that, we use tidyverse, which we loaded earlier, and deploy a function we call filter. So when we load when we load the library tidyverse, it comes with a number of packages. One of them is deploy, and within deploy package, we have this filter function which can help us to only pick these observations, the rows having human observation, living specimen, and preserved specimen as the basis of record. So I don't want to use unknown. I don't want to use material sample. So I can run this. And then I can now check the number of rows. I call it Gravia observed. So I can check the number of rows. They have reduced to 593 because I've left out, what is the sum of this? You can sum up that and subtract from the original 600. There are 7, unknown and material sample. 3 plus 4 is 7, from 600 are 593. The number of columns are still 146. So there are so many columns in GBIF database, in GBIF data sets. So what we need from there are just the location, latitude and longitude data. If I check Gravia observed, and then I use this $sign in R, it helps me to see all these variables, 146 of them. Yeah, there are so many. I won't need all of them. So the one which I will need is lat. So lat is here. This is for latitude, and then I will also need lon. So there is also longitude column or variable. So those are what I need. So I use select. I don't use filter function. Select is also coming from dplyr. Plus this.
Oh, actually,
sorry.
Oh no! I just wanted to add it on to someone who's not familiar with the percent arrow, and that is like doing and then. So for example, you're grabbing the Gravia observed data,
yeah,
and then you are selecting the longitude and latitude.
Correct.
Yeah.
Yeah. So this is just and then. Wake up and then wash your face and then take a cup of tea and then go to school and then read and then write and then come back. So it's an and then kind of thing. So, or then, if and it's not supposed to be there, you can say then, do this then. So pick Gravia observed database or data frame, and then select longitude and latitude, as simple as such. So if we run that, and then we check the head of this, sorry, and then we check the head of the data, we'll see that our data set is only now having longitude and latitude records. So all other 144 records are left out. Yeah, so a very powerful package, function. I like select, I like filter a lot. Again, dimensions are now 593 and 2. Yeah, rows and columns, 593 rows and 2 columns, longitude, latitude. Then column names, of course, lonlat. Call names function is in base R, dim base R, head base R. I don't need to install them. I don't need to install any package to see the head of a data frame or an R object. Adding species presence variable to the data frame. So our data frame now is only having longitude and latitude, but then we need another column here for the species. And we know these are locations where people, scientists across the globe, botanists, saw this species. So we can say in our Gravia coordinates, which we created having these two variables, we add a species column. Adding a column to a data frame is very simple, just $sign and then the name of the column. So Gravia coordinates, $sign species, and then assign 1 to that new variable you create. Yeah, so just that line of code is enough for that. And if we check the head now, we'll see that we have longitude and latitude as well as the species column. So This my data set. Sorry, let me check the tail. This my data set. Tail of Gravia codes has got NA. There are some locations where people collect or recorded the species that it is here, but they didn't have GPS. They couldn't locate it precisely in terms of longitude and latitude. Or they didn't have a way, or they did not record that and forward to the database. So these NAs we cannot know. We don't know where these latitudes and longitudes are. So we need to remove those. We can't model an occurrence where we don't know that it was there. So removing NAs from latitude and longitude, there are many ways of removing NAs in R. You can use NA omit, you can use drop NA. A serious programmer can also create things like if or while, or those kinds of iterations to throw away NS and remain with only where values are present. But in R, we make things simple. You just drop or you omit. So drop NA or omit NA will help us to do that. I don't want to use omit NA because if we omit NA, then for example, this observation 588 will go away, but the next one will be 589. So the listing or the row names will remain the same, but the observation will be gone. But if we use drop NA, then it throws away all the NAs and leaves me with new row names, which is assigning from one to the last one, which is more preferable to me. So again, and then take my Gravia with coordinates and then drop NAs. So it throws this data frame into this function. I don't need to rewrite the data again here. So it throws it here and then removes all those observations with NS. Having the data as spatial point data frame or geodatabase geodata frame or geopandas geodata frame or something like that, or having it as a spatial points data frame, I just say, I bring in the coordinates function and throw in the data into it. I could have used data and then coordinates function and then pick the longitude and latitude. But what I'm doing here is that I'm assigning longitude and latitudes columns in my data frame now. And then I'm picking those one and then assigning them as coordinates to my data frame. Just before that, if we check the head of Gravia complete, or is it head? Yeah, head of Gravia complete. Not really head, but number of rows. Sorry for that. If we check number of rows, there are now 246. So we had a lot of information that were just NS in our data frame with lat long. Now we are down to 246 occurrence records that are complete, that have got latitude and longitude records in place. So let me assign the coordinates. And then if we check this, the class, it is now not a data frame, but it is now spatial points data frame. As simple as such. The next step, now I'm satisfied that I have my occurrence records that are clean. They have all the coordinates. They are. I've also assigned my species variable and then set the latitude and longitude as spatial points. Then I get climate data. Get data function is helping me to get world clim data, and the variable inside the world clim data that I'm interested in is bio. Then the resolution, I am interested in 10. Bio are bioclimatic variables in world clim database, and they are 19 in number, from bio 1 to bio 19. Resolution are from 10 arc minutes to 30 seconds. So like 10 arc minutes are 18 kilometer, around 18 kilometer resolution. So 18 by 18, a very big pixel. If it's coarse, let me say it's a coarse resolution or not fine. If we go to 30 seconds, arc seconds, then we have a finer resolution. I'm using this larger resolution or coarser. Let me not say larger resolution because I know resolution and all that are conflicting or confusing. Let me use coarser. I'm using this coarser resolution so that it's easier to download. It downloads faster, and then it's okay for my variables. It's okay for my extent. I'm doing modeling for the whole of Africa, so I don't need a very, very fine resolution for that. I did that, and then if I run this claim, I hope it's there. Yeah. So claim object get data function has helped me to acquire world claim data of variable bio and resolution 10. So if you just run this claim object, it throws this information to the console that this is a raster stack with dimensions 900 rows and 2,160 columns. So this is the number of cells, and there are 19 layers. That is number of layers. There are 19. So I have 19 layers of biodata, of bioclimatic data. The class, it has thrown it there. It's a raster stack because it's a collection of several rasters layers. We can plot it because it's a raster layer. We can simply plot it, but I'm only plotting the first layer. So I'm not plotting all the 19. I can plot all the 19 if I don't include that index. So all of them will be thrown on this lower right plotting section. Then I will remove that and again plot just one of them so that when I add the points, it's easier to see. I can plot the first one, I can plot the 19th one, whichever, because I know there are 19. Let me plot even the 19th one. The first one I plotted, yeah, quite not very clear. So maybe I go back to the second one, probably a bit clearer. Yeah. So there are different layers representing different environmental variables. So if I run points on my Gravia complete, then I will throw the points, the location data of the occurrences on the environmental layer I've just plotted here. So you can see they are distributed primarily in Africa. So we don't have Gravia 10x in Latin America as well as in North America. So covering exercise books in those directions might be difficult, kind of. Clipping occurrence data and cropping predictor variables to study area. You can use this draw extent. If you use this draw extent function, then if you run it, then your cursor will turn into, will hold a function which can enable you to draw a region of interest on the plot on our lower right. Do I need to do that? Let me do that. Control Enter. And then my cursor now is a plus. So this plus, I can use it to click here, and then I click on the lower right, and then it draws a bounding box within that region. I don't want to use that. I will use the extent for Africa, which we created earlier. So the first thing to clip is the Gravia complete, which are the occurrence records. I don't want to do modeling for India. I can see 2 points in India and also somewhere here in this peninsula. Probably I'm not interested in those. For now, we'll work with Africa. So earlier, probably if somebody joined later, earlier we created an extent of Africa object. So Africa extent object is X minimum, X maximum, Y minimum, Y maximum. Minimum longitude, maximum longitude, minimum latitude, maximum latitude. And then you throw that into extent function. So it's an object, and it's thrown into the my environment, so I can use it. So Gravia complete crop is now a new object. The names of the objects, I like writing them as long as possible or making them as long as possible so that I know what the object really holds. You can say gravia 1, gravia 2, gravia 3, but then you will forget what is 1, what is 3, what is 4. But if I say gravia complete crop, then I know it is gravia with complete latitude and longitude records. And again, it is cropped from the original complete set. And then I crop it using the African extent. If you are in QGIS, then probably you will use clip, because this is a vector we are clipping. So crop or mask, sometimes we use it when we are dealing with rasters in QGIS. So it will take some time. You can see this red sign saying stop. So it tells me that R is still doing the cropping of occurrence records that fall within the extent which I provided for Africa. Taking longer than expected, probably because the computer is now also on Zoom. I wish I didn't run it because I already had it. I could just go ahead. I already had it in my environment. Okay, while it's running, the next thing is to plot the claim and then plot the points again. And we'll see a difference that the Indian points will be left out. The 2 points that are in India will not feature in the next plotting of the cropped data, as expected, of course. If I click escape on my keyboard, I hope it will stop running. Yeah, with some error, but I hope since I already created it, it should be fine. Yeah, so I can plot the whole of the study area, the first layer of that, and then add the points. Yeah, the cropped points. So the 2 Indian points are gone. So I'm now satisfied that I'm using the records that are specifically located within my area of interest. Why am I omitting the Indian points? Probably based on the conceptualization stage of my study, I realized that the species is never in India. And then those occurrences in India could be due to poor entry of latitude, longitude information, or some other reasons that are not really capturing the range of the species of interest. I can also crop the climatic data using Africa extent. Yeah. Why am I doing that again? Yet I did it earlier. So again, it will take some time to run. I can abort it by using escape on my keyboard. Then I can just plot what I cropped earlier. Now you can see it's only Africa region. Part of this is showing because I didn't clip using a shapefile. If we were in QGIS, or the same could be done in R, if I had a shapefile specific for Africa, then I can just use the shapefile to clip to the perfection of Africa. But now just I used a bounding box stretched outside Africa. Then I can add my clipped points. You can see still some parts in that far right. Well, so what do I have on my right-hand side? I have climatic data, variables that are for the species of interest for me. At the same time, I have occurrence records fitting within my area of interest, Africa. So I can develop my species distribution modeling. But before that, I have 19 climatic variables. These 19 climatic variables could be correlated. Yeah, they could be correlated. Some of them, 1 and 2, 17 and 4, 16 and 3 could be correlated. So, but I don't need to run data modeling using collinear variables. So you can run this code here, the one I'm highlighting, if you have a good machine based on your study area. But because of the number of pixels in this Africa map on your right, I'm not going to run these pairs of climb crop, all of it, but I'm only going to pick 200 random pixels and run the pairs function to see which ones are highly correlated. I could do it, but it will take longer time with the first one, all the pixels. Yeah. So if I run that pairs, we see this kind of output. This one is showing us which species are highly correlated, or which variable is highly correlated with which one in terms of environmental variables from bio 1 to bio 19. So those that are collinear, like this is bio 5, is collinear with bio 10. And if you look at this correlation coefficient, it is 0.96, others 0.95. These are very high, even 0.99. Which variable is that? Bio 13 and bio 16, this one here, highly correlated. So those ones, you drop some of them. This N cell claim crop 1 is just counting the number of cells. So there are 181,000 pixels there. And then there are 19 variables. My machine will take too long to run that. So here we bring in the USDM package, which I loaded earlier. So USDM package has got this VIF step and VIF core functions, which are helping us to check for collinearity across these environmental variables and drop some and remain with some. So VIF step. Okay, the first thing we do is to extract, because we have occurrence records and we have environmental layers, then we can extract environmental values coinciding with specific occurrence points. So occurrence point X has got bio 1 value this, bio 2 value this, bio 3 value this, because those are what we are going to use to train the model. So that's, I'm going to run this. As data frame function is going to render the output of this function as a data frame. Otherwise, if I didn't use as data frame, then it will be a matrix or an array. But then again, we can change all those to a data frame. So I run that. That runs faster because it's just manipulating the value which is already. We have few extraction points. We have occurrence points that can easily extract the values faster. We are not using all the pixels, all the 181,000. No, we are only picking a few. If we check the class, no need for that. It's a data frame because we use as data frame. If we check the head, then we will see that each, we will see the output of the values that we extracted. So we've extracted values of bio 1 up to bio 19 for each occurrence record. So it is only these extracted values that we want to check for collinearity, because we cannot check collinearity for the whole of the region. We are only interested with where the species occurs, because that is where we can separate the variables to use in modeling the species distribution. So what has VIF step done? VIF step function on our current climatic data, that is where the species occurs, the climatic variable corresponding to that. It has thrown away 10 variables from the 19 input variables because they have collinearity problem. And which are those 10? They are bio 5, bio 11, bio 7, bio 1, bio 6, bio 6, bio 12, and up to bio 17. So it gives a very, very good information here that after excluding the collinear variables, the linear correlation coefficient ranges between this and this. So the maximum correlation which is left is 0.65 between bio 9 and bio 8. Threshold is sometimes, for correlation, the threshold is sometimes 0.7, but you can vary it for your need. So the remaining variables which we are going to pass into the modeling are 9 out of the 19. So I will use exclude function to remove the VIF step 10 variables which have collinearity problem. Yeah. So this does that. So This one, do I need to do that? Okay. We can plot the remaining variables. They are 9 in number, 1, 2, 3, 4, 5, 6, 7, 8, 9, but they don't follow that order. You can see it's bio 2, bio 3, bio 8. Other bios were removed. So we are only left with this 9 bios suitable for the model. So to model, to use SDM model, you need SDM data. And SDM data, you need this SDM data to prepare the occurrence data, which is the species part of the data, as a function of all the variables which we extracted, and then train using the Gravia complete crop. Yeah, so this is the occurrence records which we extracted, which we cropped for the study area. And then the predictors which we are using are the climate crop non-correlated. This one I created here when I was doing the exclude function. So non-core are those that are not correlated. So if we run this SDM data, will it take long? I think it takes long. So maybe let me see if it's safe there. Yeah. So I saved the output of that function to this SDM earlier. You will run it if you're running it for the first time, but it's good that it's already there. So then we have the number of species. Sometimes you, like I said, there is a joint species distribution modeling. You can model more than one species. So for our case, it's one. Species names, we only named it species. Then number of features are 9 because we left out 10. Those are the environmental variables and they are bio 1, bio 3, bio 8, as you can see here. Then presence background, no, okay. I would have run the other one, but no problem because the output here is for this other object. So I overwrote the first object using this other function. What we did different from the first one is to add a background. So background here is we are generating random points within our area of interest, within our area of study, where we generate 1,000 what we call pseudo absence. Absence of a species is a very important record. You can walk somewhere and say, okay, elephant is not there. You can go to Arctic and say elephants are not there. So that's an occurrence record, but it's an occurrence, it's an absence occurrence record. Very important in modeling. So we generate 1,000 of them, background. There are so many information that guide where or how do you pick these 1,000 records. We might not get into that, but there are sometimes when you can say, okay, let me pick them to the west of Africa, let me pick them to the southern Africa, and then run the model and see if there is a difference. Or let me pick them in areas where we don't have large water bodies, if they, something like that. There are so many reasons. So the next thing is to run the model, and there are so many methods. There are so many methods that are in SDM which can help us to run the models. Like I said earlier, random forest is there as a method. Support vector machines are there. GLM is there. GAM is there, and so many others. So if we get method names, if we run this empty function, then it provides a list of available methods for SDM, currently 21. So all these are methods, GLMnet, GLM, GLM GAM, GM-poly, GLM poly, all those are methods which you can use. Why or how do you use which, or when do you use which is a long process. It needs a lot of review of the method itself so that you understand what the method needs, and then the reasons why you use it. It's just like Pearson, Spearman, Kendall-Tau, and whichever correlation methods you use, you have to be specific why. So for this case, we run Gravia model, where we use a function SDM, then we provide species and all the variables, the 9 of them. Then we say data is SDM data, which we created here. So that is the data which can go into the SDM function to run the model. You have to prepare the data first. Then the methods, which I said we have 21 of them, I'm only going to pick 3. I'm picking these 3 for demonstration. I didn't read anywhere to say this species, I can only model it using these 3. But sometimes for academic, for publication, for all that, for policy, you need to support why RF is preferable to Maxent, for example. Replication, I'm going to replicate this using subsampling as a method as well as bootstrapping as a method. So I provide those 2 replication arguments. Test percentage is 30. So this, I'm going to train the data using 70% and then test with 30% partition of the data. And I'm going to replicate this 3 times. I'm seeing some drawing on my screen. Probably somebody's doing something elsewhere. No problem.
I'll say you're drawing accidentally. Oh yes, it moved.
So what we have here is that I have 3 methods times 2 replications. So those are 6, then times 3 number of replications for these replication methods. So that gives me 18 outputs. So there will be RF subsampling, there will be RF bootstrapping, 1, 2, 3 as the output objects for this species. Then if we run the model, what information can we get from the model? A lot of information. We see that the model, I didn't run the model now because it takes some time. Of course, not 1 hour, not 30 minutes, not 10 minutes, but like 3 minutes, which we don't want to waste. So the model ID are 1 up to 10. The species, we have only 1. The methods are RF up to 6 because of subsampling 3 times and bootstrapping 3 times. GLM, the same, and support vector machine, again, 6 of them. Replication, 1, 2, 3, up to 6. Then 1, 2, 3, up to 6 for each method. Success, true. So it means they ran perfectly well. Training, true, but test dependent, we didn't have that because I just partitioned the data I have. I did not create, sorry for that. I did not create, I did not have an independent test data. If we had that, we could include it and then we will see test depth. Not really test dependent, but test independent. Test dependent is true, but test independence is false. It's not independent test data. Rock curves, we can generate rock curves from this. Those who have done some modelings, these kinds of things, we understand that the best ROC curve should be closer to 1 in sensitivity axis. So output like this, GLM did not perform so well, bootstrapping GLM did not perform so well, but random forest, subsampling and bootstrapping, as you can see in the titles, performed pretty well. GLM subsampling and bootstrapping did not perform so well. Support vector machine, both bootstrapping and subsampling performed in between the 2. So that output we can generate pretty fast. But we can also smooth it. If you look at this output, it's a bit ragged. It's like staircase. It's like staircase. So we can smooth it using a function, an argument in the rock function called smooth is equal to true. They look a bit smoother. Then we can also have a GUI or a graphical user interface of the model. And the GUI is very good because from it we can run we can check a number of output, a number of interesting output from the model, yeah? Like here we can see we ran 1 species. We used 3 methods, RF, GLM, SVM. Replication was subsampling and bootstrapping, and 3 times replicates per method. And then we did 6 replicates per species because of 3 and 2 subsampling and bootstrapping. Then test percentage was 30. This is important. This information here is important because it tells you which of the models, which of the methods you used ran successfully. So 100% here is a great information. Down here, we are interested with the AUC, but also these can also be important and reported. Random forest performed the best in terms of the AUC statistic, 95%. GLM in SVM in between GLM and RF. That one we saw even in the rock curves we generated. I hinted that SVM was in between GLM and RF. Model ID, species, this is the information we got earlier. Evaluation in terms of area under the curve. We can also see that for the species. If we add more than one species, their names will be here, like 2, 3, 4, 5 of them. And then modeling method, you can pick random forest, then you will only see the output for the random forest subsampling and bootstrapping, as well as GLM can also show you only for GLM. If you want GLM and RF and SVM, all of them, then you can show all of them on the same window. Then replication, bootstrapping. If you're only interested with outputs for bootstrapping or subsampling, you can view all those. Information on calibration, information on threshold optimization will take us earlier. Threshold independent, we also have, sorry, I wanted to talk about the plots, density and box plot. Yeah, so here is also showing us all the models that we ran, 18 of them. Remember the methods are three. Replication 2 and number of replication 3. So multiplication of all that gives us 18. So we can compare the performance in terms of presence as well as absence. Then maybe we don't take too much time on that, but it's a great graphical user interface output of that run on Shiny app. Archive, we can also run archive to see the output in terms of response. It prints out response curves for the species and the variables. We have 9 of them, bio 14, bio 15, up to bio 9. And then there's a very important information because it tells the individual researcher that as bio 14 increases from 0 to 150, this is how the species responds. So clearly we can see bio 3 and bio 9 are standing out because it's telling us that for bio 3, if the values increase from 40 to 80, the response of this species is quite linear. It's quite linear. Same to bio 9, but for the difference is inverse, yeah? For bio 3, the increase in the values of bio 3 over space is supporting the response of the species. On the other hand, for bio 9, as the values of the bio 9 increases, the species decline steadily, response declines steadily. Some straight ones like bio 14, we can say that as bio 14 increases from 0 to 150, there is quite minimal response to the species. In Kenya, you will say H2E species, yeah? It doesn't mind the change in that variable. But for this variable, bio 3 and bio 9, it matters. The change matters a lot for the species. We can get a verb importance for the model. This is another important output down here, but then it will be more easier to understand if we run the plot. The plot, that is plot get var importance for Gravia model method RF. I've narrowed down to random forest for this output. So what we can see is that Bio9, in the random forest method, Bio9 stood out as very important in compared to the other 8 variables. It was the most important in contributing to the distribution of the species compared to the other 9. Bio3 also performed very, very well in terms of determining or influencing the distribution of the species over space. A very, very important output, because now you know if you are dealing with this species, then you need only to control Bio9 and Bio3. If these are pH, then you look at the soil and you do something to the soil to alter the pH to stabilize it. If this is temperatures, then you see how you control the temperature. For example, these are chicken in your house, and then you find temperature is the greatest determinant on the existence or on the occurrence, like when it is cold, the chicken behaves this way. So this is what you deal with. If it is climate change, then the policymakers know that, okay, for this species, Bio9 or this variable is the greatest. Then we can also plot another great output, which is called niche. For the niche, what it does is that it creates on a 2D, in a 2-dimensional output of selected variables in terms of their contribution to the species distribution. So for example, if we run niche function on our non-correlated variables for Gravia ensemble, which we created, did we create Gravia ensemble? Not yet. So probably I will not run that now, but I will run it later. We are not yet there, but I will run it later. Great. So at least we can see the output of our model. I ran this one for random forest, but I can remove this method for random forest and run it for the whole model. And still we will see an output, which is indicating again, Bio9 still performing great, and Bio3 follows, then Bio16 somewhere. So you can identify which environmental variables were great in that. So line 188, we'll come back to that. Now we have our model. The model we've trained using the occurrence records, 157 of them, and 1,000 pseudo absences, which we generated randomly over space. But then we need to predict now over space, over our climate crop region throughout Africa. Yeah. We now need to project this throughout Africa, that one, to see the potential distribution of the species in areas which are matching similar kind of variables that were predicted in the earlier output. So we run this Gravia predicted, which we throw in the model, Gravia model, and then the climate, the area which we want to predict, and then we save the file using a given name in our working directory. So if I run predicted, we can see it produces a raster stack, class raster stack, and then 18 layers. Why 18? Because we ran 18, we got 18 model outputs. 3 methods, two replication methods 3 times. So 3 times 2 times 3. We can get the names of the predicted output, and the names follows just that. It says ID number 1 is species 1, and the method is random forest, and replication method is subsampling. So that it does 1, 2, 3, up to the 3rd one. After this 3rd one, it goes to bootstrapping as a replication method. Still for RF. If we get to the 6th one, we are done with bootstrapping. We are done with the random forest. This first chunk is for random forest. Then the second chunk of up to 12 is about GLM. After that, we go to SVM, support vector machine. So we have 18 layers generated for the predicted areas. We can plot any random number of them. We can plot all of them, 18, which will clutter my plotting region. But let me make them 4 so that they fill well the region. Maybe another one is number 17. So if we plot those 4, we will see the output of prediction. So what we have as the X, as the legend here, tells us the probability of occurrence or the likelihood of occurrence of the species at that particular point. So we see this species being predicted in the Namib Desert, the border with Angola, part of Mozambique or Namibia, yeah, by this. But then it differs because here we can see in West Africa, it's predicted somehow there, but then if we come to this SVM, it says no to West Africa for the species. So different methods generate different outputs. Yeah. So it is important to generate an ensemble of that. Yeah. And in ensembling, what we mean here is that, let me use the word averaging. You use an average or a median of these layers which you have generated here. Yeah. Of these predictions, so that you are more confident with your output. But then which layers do you use for generating the ensemble? A number of factors can contribute there. You can submit your output layers to different experts, and then they look at them and say, okay, this model is totally wrong. You will never see this species over this region. They can also say, okay, this one is good. This one also performed good. This one also performed good. You can also dig into the literature and see the potential distribution of the species or the range of the species, IUCN databases, and so many other sources where you can then narrow down and look at the output and say, okay, this one and that one performed well. This is in addition to the output we saw earlier, which was telling us the performance of each model. That model 1 performed better than model 2 and all that. Where we, let me just go back to the GUI. Okay, the GUI indicated that, maybe that will take more time. So for example, random forest, I said performed better. And I know that from ID number 1 of our model to number 6, those are all random forest. Yeah, they are random forest. The first 3 are subsampling and the second 3 are bootstrapping. Yeah, so if that performed well, and expert opinion also indicate that the species was well predicted with the random forest other than all these other methods, then I'm more confident in presenting an ensemble using that method. When evaluating my model with the expert information, I don't need to tell them that this was generated by random forest, this was generated by GLM, this was generated by Maxent. Because different people will say, okay, since I'm the author of RF, I must support it. Or because I'm interested, all along it is known to be good, then it is good, something like that. So you hide that information and then you just let them evaluate based on the outputs you have. So Gravia ensemble, if I run that, it will just be a raster layer. It will not be a raster stack because I've ensembled 6 random forest output layers. You can pick any 1, 2, 1, 7, 1, 8, 10, all of them. You can even ensemble all of them. Yeah. The names of this ensemble will not be 18 like earlier, but it will just be one Gravia ensemble. So we can also plot it. It will just be a single output, not 18, just a single output. Now, I want to create a color ramp because we can see the spread here. It's not easy to using this base color. It might not be easy to tell which regions are the species occurring and which ones are not. So color and palette is helping me to pick some colors, white, green, red. Sorry for those who are very, very good at colors. I might not be one of the best in colors. So I have generated an object holding, actually a function holding the colors for me. So if I plot ensemble using that color, which I just saved in that function, then the output is different. Probably a number of you are saying, yay, that is nice. Some of you are saying, yuck. That's another topic of discussion. Ladies, those colors, I don't know whether that dress will be nice or something. This is my ensemble output. I can throw it to map view. Yeah, I can throw it to map view. Simply the ensemble and then I color and throw it to map view function. Map view will take some time, throw some information there, but in the end, give me the output. So the beauty of map view is that you have so many layers underneath your plot, and then you can zoom in to an area and then check, is this mountainous area? Is this a lake? Is this a what? Why is this species being better predicted here than any other place? Yeah, so interactive nature of the map view is so powerful. And then as you move the cursor over the region, you see the latitude and longitude values are displayed here. Very, very tiny, I'm sorry for that. But at the same time, you are able to see the pixel values, the prediction or the probability values changing up there. So as you move in this red region, it's 0.7, 0.8, 0.5. But then as you go to these light areas, it's 0.0 something. So least likely occurrence of the species. We can also add the observation, the observed data. We still have them on our plot. So if I run this, we will plot the occurrence records on top of our layer. Yeah, you can visualize them and then compare with what it is predicting based on that. We might also, okay, what we are seeing here is a range from 0 to 0.9, 0.1, 0.2 up to 0.9 and probably 1. So you might ask yourself, where is the species occurrence? Where is the species present now? Yeah, where is the species present? Because it's 0.4. Does 0.4 mean it is there or it is not there? Does 0.9 means it is there or not there? So what we do is thresholding. It's programmers will say if else. If the pixel value is below this value, then absent. If the pixel value is above this threshold, then present. Yeah, as simple as such. But then for this, we'll bring back our data, which was SDM data, to a data frame first, using as data frame function. You can check the head, no problem with that. It's a data frame, species by 1 up to 19. Of course, leaving out other bios which we eliminated. Then we provide the coordinates to the data frame so that our values which were used in modeling, these occurrence values, have got the coordinates from the SDM data itself. Because here we don't see the coordinates in this data frame, the first data frame. So if we run this, then it will throw in the data frame and also remain only with the species occurrence record. So it is species and latitude and longitude data. So this one is enough for us to pick, to extract pixel values and then use some if-else function. We'll run an evaluation, first of all, which will tell us, which will help us to pick the threshold to query the predicted layer so that we have values below or above this to run the presence absence. So if we check evaluation, it gives a lot of information, TSS, AUC, specificity, sensitivity, TSS. These are statistics values or statistically generated values which can help us do the thresholding. So the threshold here is saying, if we have 2.8, or this is 0.28 and above, because the maximum here is 1. So if we have 0.28 and above, then that is presence. Below 0.28 is an absence. This is super strong, yeah? This is super strong because sometimes you can have it to be 0.6, 0.7, 0.8, but here we find 0.2 because the method we used, which was random forest, really did some good job in predicting the species. So we will pick this second row because it maximizes on TSS. That's a deeper kind of detail on that. You might get it in other sources, or we can explain it later in another session. So we can create a threshold object, or we can just say 2. Yeah, we can create a threshold object, or just say 2, the second one. Not really 2, but negative 0.28. We can just pick this value and throw it here in the if-else function. But I will create the threshold and say it's picked from evaluation. Sometimes in R, we use @ and sometimes we use $, depending on the object, the data object type. So we have already picked our threshold, the second one, which is negative 0.28 we saw earlier here. Not negative, but 0.28, because it is 2.8 times 10 negative 1. So this decimal point moves to the right once. Classified, we create an empty raster where we want to throw in the values we generate here. We create an empty raster using the raster function, and the raster we create should be the same as the ensemble, should take the properties of the ensemble, the resolution, the extent, and all that information. Then we fill in, because it's empty. If we do it, if we check classified, if we just run classified like this, sorry. Yeah, if we check the classified like that, it's a raster layer. It has got those pixels. Resolution is that one. Extent is that. Projected to that, but no values. It has got no values, minimum, maximum, they are not there. It's empty. So we want to feed in data from the if-else, from the ensemble, Crevia ensemble, which is the output we have here. We want to extract data there using if-else function. So if from that layer, we see a value, a pixel value greater than the threshold, greater than 0.28, then assign it to 1. Otherwise, 0. So we get classified field. And if we again check our classified, then we will see, sorry, if we check classified down here, we'll see that now it has got a name, names one layer, then values 0 to 1, minimum, maximum. So it has been fed with the values from the if-else function, which has run through this model, this layer, and picked what satisfies that. So we can plot our classified output. Yeah. So the plot here is now telling us that this dark green, the color now is a single color kind of. So these are where the species is present. This is up for debate. Yeah, a lot of queries need to go in here to say that the species is actually there. But these are the regions that are suitable for the species to exist, to occur. The condition here is suitable. But then if you go to the deeper details with the experts, then they will tell you, okay, here it is suitable, yes, but this is a town. This is the capital city of this country. Yeah, we will find one in somebody's backyard, but not throughout this area, yeah? So something like that. Those are layers which can be fed in and then you revisit your output. But then there are so many things which have gone into this. One, we have only picked random forest. Remember, we modeled using SVM and also GLM. We've thrown those away and only used RF. We must justify why we used that. Yeah. So that somebody who is interpreting this, somebody who is saying, okay, this is a suitable region for this species. Let me start planting it and see if it will grow. Yeah, something like that. I've only done for the present. This can also be done for the future. If you have a climate layer or projections into the future, like IPCC representative concentration pathways, the 2 point something, 5 point something, 4 point something for the future, 70, 2070 and all that, you can make predictions using your trained model on that data and then visualize the potential change. Like you will see that the species will spread towards the north or to the south or the west or to the east of one specific location. At the same time, you can use this model if, for example, we didn't have occurrence records in the US, we didn't have it in South America. So you can use this model with a different layer in US to predict the potential occurrence of the species elsewhere outside your study region. Transfer of such model is a big process, and it is what people, what researchers use to predict the potential spread of a certain invasive species. Like if it has invaded this region, then you say, okay, the other region elsewhere has got the same climatic conditions like that place, soil conditions, whichever conditions, then you predict that it's likely to spread to that region, like the stubborn prosopis in northwestern Kenya. There was something we left in line 188 because we had not created the color ramp. It's called the niche. So niche, if we run this, it won't take too long, I believe. If we run niche, what niche does is that it picks two variables. It feeds into variables so that we have a two-dimensional output of the niche to tell us which regions within Bio 9 and which regions within Bio 16 are supporting better occurrence or distribution of the species. So for example, here we see 151 to around 1 or 200 for Bio 1, Bio 9, and then just above 100 or somewhere there for Bio 16 are the highest or gives the best conditions for this, the occurrence of this species, dark red region, or 0.75, 0.8. So these are better. This is helping you to know that if you have another place elsewhere, which Bio 9 reads within this range and Bio 16 reads within that range, then the species is likely to occur in that region. If you plant it there, it's likely to grow. But then that is subject to so many other variables, so many other functions, so many other assumptions to be made before you arrive at that. I think that's all I had for today. Predictions into the future, running joint species distribution modeling. We can organize that if some people are interested on a later date. But this is I like it. I like doing it in R because R is, as you can see, is user-friendly. We've not gone into any deep coding where somebody gets confused or something. Everything is straightforward, like using FXMS calculator in Form 4. So nothing is too deep. But before we run this, there are a lot of assumptions, a lot of readings which go into this so that you understand what you're doing. For me, I'm modeling wild edible plants in Turkana, Kenya. So a similar work to this is what I do. I get occurrence data, occurrence records for those species, and then I pick environmental variables from so many sources, see which ones are better in contributing to their distribution, and then run my models at the moment. So, Brian, I think that's all I had for today. Thank you, everyone, for your attention.
Yeah, sure. Thanks, man. It's an amazing domain, and yeah, it was really great insights. I believe yeah, people have gained and are learning about it. Yeah, so how did you start to use that? Because maybe people think you knew this from the start.
Yeah, great. I started learning R in 2018. I was an intern by then at Calro, the Research Institute in Kericho, where I am at the moment. But I'm not at the institute. I'm no
Yeah,
longer at the institute, but I used to be there. So when I was there,
yeah.
a student came from Chuka University. She's called Sharon Cherotich. So she told me, okay, Wycliffe, why are you using MSTAT? Why are you using Stata? Why are you using this? Why are you using this? Why are you starting the same process again and again? When yield data from T fields come, we run ANOVA. We were running ANOVA in MSTAT package, statistical
Yeah.
package. So she told me, Wycliffe, this is too tiresome, yeah? Every time somebody comes, replicates whatever variables, we were entering these things and she was like, okay, there is something called R. You can try it. So she introduced me to R, and then when I started doing my master's, I had to read a lot of publications here and there. And then I came across some papers. They say, okay, field data were collected using this, analysis was done using this package, and this program, plotting was done using this program, writing or editing was done using this program, and then published. So that's a paper which can take somebody too long to reproduce. But then when I read some ecology papers, in ecology letters or in ecography or in evolutionary ecology or such like journals, I could see somebody saying, okay, all analyses were done in R, as simple as such. So I said, okay, this is an interesting area. And then when I saw the plots generated in R, they were so beautiful. And I said, okay, let me dig deeper into R. So when I went to Germany 2019, I also had an opportunity to interact with R because it was part of the course for the ecology students or ecology and natural resource students. So that was when I also got a better uh, uh, grasp of the package or of the program.
Yeah.
Since then, I've not looked back. I just work with R a lot. GeoPandas came in Python. I also tried it a bit, but then I saw SF could also do similar things. So I said, okay, I know how to use it in GeoPandas. I know how to use it in R. If job calls for each, I can run them.
Yeah, sure. Definitely, when you have both and you're into data science, yeah, you become unlimited. You can do so much, too much that yeah, without needing a license, and it's really great. And it's usually about automation. So
Yeah.
yeah, we just encourage everyone to keep planning and knowing what you really want to do with your career and stuff. And yeah, you can learn this, and resources are out here for free. So it's about an individual's effort.
Yeah.
Yeah, we can show, but yeah, it's up to an individual to just dig in
Yeah, sure.
and pump in the time. Yeah, that was super wonderful. Yeah, I've seen someone has commented on the chat too. Wonderful session. Thank you so much, Wycliffe.
Faith Mushembe, thank you.
Yeah, so yeah, we can call it a day. Maybe next time we can do about future predictions if people are interested in this machine learning stuff. So it's fine.
Yeah.
This was actually so great. Yeah. Okay, so I'll end the session. I think it was great. Unless anyone has got burning issue before we close.
Maybe you also include that we will provide the slides and also.
Oh yes, yes, and also the
script.
scripts. The scripts will
Yeah,
be
the script.
shared. So, yeah, so that they play around with. Yeah, sure, definitely. Okay, then that's it. Bye, everyone.
Bye.
Bye.
Goodbye.